Okay, so in 21.6, we're going to start talking about the structures. There are 4 different structures that amino acids can have: primary, secondary, tertiary, and quaternary. Primary, secondary, tertiary, and quaternary. We're going to go through each of these separately. So first, we're going to talk about the primary structure. All right, so we'll talk about the primary structure. Pretty straightforward, very easy. The primary structure is just basically the sequence of the amino acids. So what's my sequence? What's the R group at my N terminus? What's the next R group? What's the next R group? What's the next R group? So for example, we can have, alanine hooked to a phenylalanine, hooked to a proline, hooked to a glycine, and then maybe another glycine, and then another phenylalanine, and so forth and so on. This is basically what the primary structure is. It's just the sequence in order of the amino acids in the peptide or the protein. Okay. Usually you also show it in this zigzag model. That's a backbone bone. So if you can see, here's one of the amino acids. Again, nitrogen, carbonyl, carbon. Here's the other amino acid, just to kind of show you where the amino acids are. Here's another one. And if you notice, it alternates. R group here, R group there. Okay, and then so here's a carbonyl, so a nitrogen would be attached here to another carbon with an R group, to a carbonyl, to a nitrogen, to another carbon with an R group, and you notice it alternates R down, R up, R down, R up. All right, it's just kind of showing you the structure of it, but pretty much the primary is just the sequence of the amino acids. All right, the secondary structure, once we get to the secondary, it's a little bit more interesting. It's not the sequence anymore, it's the 3-dimensional arrangement of these amino acids. This is the most important part about proteins. All right, how proteins work is really not the sequence of the amino acids, it's the shape, it's the 3-dimensional structure it makes. Does it make like a football shape or a spherical shape or a square shape, or like what kind of shape is it? Because the way our bodies work is what's called a lock and key model, where a substrate, whatever it is, binds into our amino acids, into our proteins or enzymes specifically, and all depending on the structure, it actually binds onto a certain thing in our bodies. It's very structure specific. All right, and these structures are based on the primary structure, on the secondary and the tertiary, and hydrogen bonding, and that sulfur-sulfur bond linkage that I talked about. So that's kind of why it's important to talk about the structures of these proteins, because they tell us something about how they work inside of our bodies. All right. So there are basically 3 types of secondary structures. There's what's called an alpha helix, a beta pleated sheet, and then a random, where it's not an alpha helix or a beta pleated sheet. So let's first talk about these shapes. An alpha helix has this spiral effect to it. All right, if you kind of see, it has a spiral. So it's like a coil. All right, a perfect example is a coil effect. It's like that. All right, it's not really shown that great here, but that's basically on the ribbon example, it actually shows you. Okay, and you're basically held all together. All of these coils are held together by hydrogen bonding. These dotted lines right here is hydrogen bonding, and the hydrogen bonding comes from a nitrogen. You can see there a blue nitrogen with a hydrogen, and the carbonyl of the amino acids. So you'll have, a nitrogen with a hydrogen hanging off, and you'll have a carbonyl hanging off, and you'll have a hydrogen bond. And you have a whole bunch of these all throughout the whole entire alpha helix. Okay, so you can kind of see all of these hydrogen bonds. So hydrogen bonding with the NH and the carbonyls is the reason for an alpha helix. And notice, where are all the R groups? Notice all the R groups are sticking outside. Okay, so it's really all of the peptide bonds, all of the stuff in the middle, which is actually all hooked all together, making these hydrogen bonds, giving this alpha helix or coil look to it. Okay, beta pleated sheet is similar. It does form hydrogen bonding, but it's not a coil. How this basically works, let me see. Yeah, I guess it's not that great of an example, this one that they're giving, but it's basically, you've got a chain of amino acids that are going this way. You kind of have a bend in the protein, and then you have a chain that's going this way, and then a bend, and then a chain that's going this way, and you have hydrogen bonding this way. So it's not a coil, it's a flat a flat sheet of these amino acids. And again, it's hydrogen bonding. All right, and again, it's a NH from one side and a carbonyl on the other. You can see an NH here and a carbonyl. You kind of see that NH carbonyl, NH carbonyl, NH carbonyl. And that's got a beta pleated sheet. All right, this is kind of the way that people show proteins. Rather than people don't really show a primary structure unless people are interested in one amino acids in the order sequence. They care more about the structure of it. Okay, so here's a alpha helix, and the sheet is kind of showing the direction. So if here is the N terminus side, so my sheet is going that way, and then if I have a bend, it's going this way, and then if I have a bend again, it's going that way up to my C terminus. And then a random is just kind of that ribbon drawing. All right, so here's a ball and stick model of a protein. I don't know what protein it is, but you can see it's very complicated. But if you really analyze, say like right now, you're like, that just looks like a mess to me, bunch of carbons and nitrogens and all that stuff. All right. However, when they write it into this, it shows you it's more ordered, I guess. All right. So all the green coils are all of the alpha helix. There's a whole bunch of alpha helix, and you kind of see it at the top end of the protein. Okay. Then you have beta pleated sheet towards the bottom of the protein. All right, so you can kind of see a little bit more of this, and then a whole bunch of random stuff. It's not an alpha helix or a beta plate or a beta pleated sheet. All right, so that's kind of what's called a ribbon drawing, showing all of the secondary structures. Okay, and here's just an example of silk in spiderweb. The regions of beta pleated sheets and alpha helixes. You can see like you have a whole bunch of it right in the middle here and a bunch of alpha helix on the outside, and it's random. Okay. All right, let's talk about tertiary and quaternary because that gets a little bit more interesting. Tertiary structure is the 3D structure adopted by the entire peptide, the whole entire thing, rather than looking at just the sequence, which is the primary, remember the primary sequence, or the secondary. What's the alpha helix? Where's the alpha helix or the beta pleated sheet at random? This is kind of looking at the whole entire thing. Okay. To maximize hydrogen bonding with water, the non-polar side chains are stabilized by London dispersion forces. So if you have like an alanine, which is, or a glycine, which are just a bunch of very non-polar groups, they kind of interact with other non-polar groups. And polar groups interact with other polar groups or hydrogen bonding. Electrostatic groups hang out with electrostatic groups, and disulfide bonds form a disulfide bond. So here's a formation of a disulfide bond where we talked about where you have two cysteines, a cysteine here and a cysteine here. All right. When you oxidize it, you get rid of these hydrogens and the sulfur bonds. So here's kind of an example of a disulfide bridge. All right. Now it could be within the same molecule, or you have two separate proteins and you form a disulfide bridge. So here is between two of the same chain, like how we saw in the example of the oxytocin. All right, in the oxytocin, let's go back real quick. Hopefully I can do it quick enough to show you. Oh, did I skip it? No, here's the structure. All right, oxytocin in vasopressin has a sulfur-sulfur bond within the molecule. But imagine an oxytocin hooked up onto a vasopressin. So sulfur hooked onto another sulfur. That would be an example when you have two different molecules forming a disulfide bridge. Okay, so here's kind of showing you now that more of a tertiary structure. It's being very specific. It's showing you a nonpolar group is interacting with another nonpolar group. Okay, through London dispersion forces. Okay, so it's not only showing a beta pleated sheet or an alpha helix, but showing you why you kind of have this kink in the protein because you have because this region is implying it's very nonpolar. Okay, here you'll have a hydrogen bond. Here you have an electrostatic interaction because you'll have a plus charge here and a minus charge there. So these two opposite charges attract each other. That's probably why we have this linkage or a bend right there. Same thing here, nonpolar with nonpolar, London dispersion, disulfide bridge, two sulfurs hooked together, hydrogen bonding. This is more of the tertiary structure. It goes a little bit more into it. It's not just showing you alpha helix with hydrogen bonding, but it's explaining why is there a bend right here? Well, because we have a big area of nonpolar amino acids. Why is there a bend right here? Because we have a little bit of hydrogen bonding. Why is there a bend here? We have an electrostatic interaction. It's not by chance. I mean, our bodies code for having a positively charged amino acid here and a negatively charged here for a reason, because in order for this protein to work, you need these two amino acids. All right, maybe it doesn't really matter what amino acids I have here, but as long as I have these two, it's important. And if our bodies stop coding for these, then this protein can't function the actual way that it can function because the shape of it is not correct anymore. Okay, and this happens with age. Our cells, our DNA over time, it doesn't get, it's not all coded perfectly every single time. All right, it's a very slow process where eventually our coding for our proteins aren't quite what they used to be. That's what aging is. When our skin wrinkles and it's not as flexible, that's because our proteins in our skin don't have all these correct amino acids anymore. Maybe it lost a few of these amino acids, and I don't have a fold here anymore, so it might be straight. So because of that, the protein is not as strong or what it used to be back when you were younger. And we'll talk about that in later chapters when we talk about our DNA and how it codes for these amino acids and these proteins. That's just kind of a heads up. All right. A quaternary structure now is when you have one tertiary structure, that's a tertiary structure, is hooked up to another tertiary structure. So when you have two tertiary structures hooked onto each other, that's what's called the quaternary structure. All right, so the structure of the protein that is adopted when two or more folded polyprotons come together. Two or more folded polypeptide chains come together. Okay, insulin is an example of this. All right, you have one protein which is 21 amino acids long, another one which is 30 amino acids long, and they are hooked together by two disulfide bonds. All right, if I were to cleave these bonds, I would have two separate proteins. But because of these two, they hook all together and they fold. Here I have an internal one. All right, so I have insulin. So notice I have a primary structure. If you notice, it's showing me a primary structure: glycine, isoleucine, valine. It's showing me the order, but then it's also showing me how it's hooked onto another chain. So it kind of shows me a quaternary structure. Okay. So here's just kind of a review of it. All right, primary structure just shows me what my R groups are. It doesn't tell me anything else. Secondary structure now starts showing me the alpha helices and the beta pleated sheet. Tertiary structure shows me more of the overall structure of it. All right, shows me all the kinks and all the bends and all the alpha helices. You know, it shows me that maybe I have N plus here and an O minus here and has an interaction. And the quaternary is where I have this hooked onto another one that's hooked onto another one that's hooked onto another one. Okay, so hopefully that makes sense. All right, so let's now focus on the human body and how these structures play a role in certain things in our bodies, like a fibrous protein or a globular protein. All right, so proteins are generally classified according to the three-dimensional structures. All right, so a fibrous protein are composed of long linear chains that are bundled all together to form rods and sheets. So it's more of a beta pleated sheet. All right, as far as fiber, and if you think about it, it makes sense. Fibers usually are layers of things packed on top of each other. All right, that's kind of what, if you say something is fibrous. So if we have a bunch of beta pleated sheets, that's basically what this is implying. All right, these proteins are usually very insoluble. So like our hair usually has this, nails usually have its skin. These are more of those types of things. Have a structural role. All right, our skin definitely structural. Our bones, all of the tendons in our joints. Then you have globular proteins, which are coiled, so more alpha helix, are compact in shape, and are water soluble. These are more of our enzymes. Our enzymes have to be all dissolved in our blood or in our cells in order to do its chemistry. So more of the enzymes are globular, more of the structural are more of this fibrous or beta-pleated sheets. All right. Enzymes and transport proteins are globular because they dissolve in our blood. Okay. So keratin, which is responsible for hair and skin and nails, and in case in other animals, wool and hooves, these are more of the beta-pleated sheet, more of the fibrous. All right, they're made up of two mainly alpha helix chains coiled all around each other in order to make a superhelix. So I just said a beta pleated sheet. I forgot about this, where it's kind of a coil of alpha helices. So it's kind of a beta pleated sheet of alpha helices. So I have an alpha helix, all right, and then another alpha helix on top of that, and then another alpha helix on top of that, and another one on top of that. So I have a bunch of these all on top of each other, which we call a superhelix. All right, these coils wind around each other in order to make stronger structures like hair. It also makes it flexible. Our alpha helixes is like a coil, so it can spring up and down, up and down. Well, our hair is actually pretty flexible. All right. Collagen is just 3 chains of superhelices. Vitamin C helps stabilize these chains, and when it's missing, you have a poorly formed collagen fiber. So why vitamin C so important? So here is keratin, just kind of give you an example, alpha helix coiled on top of another alpha helix coiled on another one. All right, here's collagen, which is 3 alpha helices bind onto each other. So it's a coil of coils. You can look at it like that. Myoglobin and hemoglobin, kind of a very well-studied protein. Hemoglobin is actually made up of myoglobin molecules and something that's called a heme group. All right, so both hemoglobin and myoglobin are globular conjugated proteins, meaning that they dissolve in our blood, meaning that they contain both protein and non-protein. This conjugated means non-protein components, which is the iron and the heme group. It's an organic molecule, which we will look at the structure pretty shortly. The iron binds to the heme group and then it absorbs oxygen onto it, and then it goes into our bodies. It transports the oxygen into the myoglobin. So the hemoglobin, I'll put H, has the iron and oxygen to it. All right, and it gets the oxygen, and then let's say here I have a muscle, which is very fibrous. Inside of here, I have a myoglobin, and then the hemoglobin gives the myoglobin the oxygen, it then absorbs the oxygen, and then the oxygen is able to actually go into our muscles. Okay, here's the heme group. All right, this is what the heme, what's called the conjugated protein, the non-protein part of it. Okay, and the iron sits right in the middle of it. All right, that's why it's important to have iron in your diet, because you need iron for your hemoglobin, which oxygenates your blood. All right, here's myoglobin, 153 amino acids, one peptide chain. You can see it has a lot of alpha helix in it. And the heme group, you can see the heme group just kind of sits right in the middle of it or on the top of it. Hemoglobin is 4 different polypeptide chains. All right, you can see a blue one here, another blue one here, a red one, and a red one, and they each have a heme group inside of it. Okay, so for every hemoglobin, one hemoglobin, Hg, all right, there are 4 different chains, 4 chains, all right, or 4 heme groups. All right, so that's the quaternary structure that you have 4 groups attached to it. Carbon monoxide poisoning, just to kind of show you what this is. Carbon monoxide has this shape to it, or structure. Oxygen has this shape to it. Notice they are identical in shape. They are linear molecules. The only difference is this has a carbon to it. Well, this carbon actually has a negative charge on it. All right, this oxygen is neutral. All right, when carbon monoxide is almost the same size, same shape, same everything as oxygen. When we breathe in carbon monoxide, this negative charge binds to the iron in our heme groups, and it binds 200 times stronger because it has a negative charge. So when you inhale carbon monoxide, all of your heme groups are getting that CO bounded to it and none of the oxygen. all right? And then when it goes out into your body, it releases its CO, or it doesn't release anything, and your body suffocates because it can't get oxygen, all right? Because the CO2 doesn't work the way that O2 works in our body, nor will it even release it. And it's bonded very strongly, all right? So hemoglobin complex to CO cannot carry oxygen, and the cells die because of the lack of oxygen. Now, in order to treat for this, what people usually do, it okay, so uh, like I was saying, in order to treat it, you usually give the patient oxygen. So what happens is you swamp the body with 100% oxygen, or a very large amount of oxygen. So it helps in trying to kick out all of the CO molecules. All right. All right, sickle cell anemia is a disorder where there is a single amino acid that is not correct in the sequence. And what it does is it puts a kink in the shape of blood. So blood has kind of this ovalish kind of look to it. It has like a dent in the middle. When you have one amino acid incorrect, um, the blood cells usually have an irregular shape and have sharp edges on it, and it hurts a lot in the patient. So just based off of one amino acid, it changes its whole shape and structure of the protein, and you can't really do anything about it. So it says that it has these elongated crescent shapes to it. They rupture the capillaries because our capillaries are very, very small, and if your cells have sharp edges on it, it rips them. It's painful, and eventually it can cause death. Okay, so let's stop right there, and when we pick it up, we'll start at the protein hydrolysis and denaturization.